So welcome to Biobanking for Data Science, which is our first lecture. And this lecture and this module, I don't think is going to be, I believe is nowhere else where you're going to find it. And hopefully at the end of this lecture, you probably agree with me or disagree with me. But I designed this module as the differentiating aspects factor in terms of what this MSc AI and digital health has to offer. You'll see that all of the other lectures are shared with other MSCs, except for the projects, which is one-to-one. And hopefully this is kind of the differentiating factor that you're going to get that is exclusive. So there's going to be a little bit of many different things that you're going to see here. So we're going to have things to do with data science. We're going to have things to do with biobanking design. We're going to have things to do with ethics. We're going to have things to do with coding. So it's going to be very, very diverse, the type of materials that we're going to see here. So why biobanking? So I would say that biomedical research is basically based nowadays on big data sets from specific cohorts that are being used and analyzed informatically to generate statistical patterns that then are applied for drug discovery, biological mechanistic understanding, and development of therapies. So biobanks are critical for personalized medicine. You're going to see me say things like precision medicine, personalized medicine, etc. There are slightly different things. So personalized medicine is targets that are specific for the individual. Precision medicine targets groups of individuals that share a common feature that is used for diagnostics or therapeutics. We're going to look very, very into detail the UK Biobank, which is the leading compendium of biomedical research worldwide. And you will see the impact that the UK Biobank has on research later on in my second presentation. So what's biobanking? Really, it's about collecting, storing, managing biological samples, tissues, DNA, with the goal of providing a resource for research. And the idea is striking a balance where you have maximum access to research while preserving the identity and the consent forms from the patients from which data was collected in order to honor and fulfill the specific purposes for which those samples were collected. That's where the ethical aspect comes in. So the collection of specimens has happened, since the centuries, but it's only become formalized really through the Human Genome Project, which was started at the end of the 90s And right now, with the advent of digital tools and AI, they become the base for research development. I'm gonna skip through that. There's a lot of emphasis on standardization. So if you collect samples, you want to make sure that the variance that you observe in the data is the consequence of biological differences rather than technical or lab differences or artifacts that have been generated because you are using a different machine or a different lab person, or it's done in a different lab. So there's a massive issue trying to make sure that those differences that are artificial are minimized. We can't get rid of them, but we need to minimize them, okay? Because otherwise you are going to end up having spurious results. So after the 2000s, technologies were developed. You've heard of high throughput sequencing, for instance. There are technologies out there that now make the real bottleneck not the production of data, but the actual interpretation of the data. And this is where we are at. Right now, whereas before the 2000s, really, data was scarce, and the real problem was lack of data. Now the real issue is how do we access data and you know, how do we actually interpret the data? That's why in biomedical sciences, I believe that the real demand for skills are around the data analysis. And this MSc course is all about data analysis. Hopefully you've gathered that already. Then in the 2010s, we started having next generation sequencing, data-driven insights, and integration with research networks, where biobanks become integral components of global research networks. As this is developing, there's increased attention on the ethical implications of biobanking, because as data silos, as data volumes are being produced, like what you've seen in AI, you know, biases, and you will be able to model better the use cases that reflect the underlying data you're using. So if the vast majority of the data comes from white, wealthy, Northern European ancestry individuals, the models that come from those data resources are going to work really well for these kind of populations. And the more different you are from those populations, the less precise, the less well the different models are going to operate. But now we are in the AI age, where we use machine learning. Do you know what machine learning is? Is there anyone who doesn't know what machine learning is? What's machine learning? Okay. Catherine, you were going to say something? Okay. Okay. Machine learning is about finding patterns, learning things about data automatically using algorithms, machines. Would you agree with that, Magda? Okay. You all agree with me too much. I'd prefer if you challenge me a bit more. So there are different types of biobanks. We have population-based biobanks, like the UK Biobank. There's also disease-oriented biobanks, like for instance, biobanks on cancer, diabetes, for targeted research. There's genetic biobanks, whose focus is on genetic and genomic data. What's the difference between genetics and genomics? Tell me. And genetics? One gene. Okay, good. So population-based biobanks, they collect, store biological samples from a broad population-wide base without initial bias towards any particular disease. The objective is to enable research in a wide range of conditions, focusing on public health and epidemiology. So they may include genetic material, clinical information, lifestyle data, environmental exposure in a longitudinal manner. So what's a longitudinal study? Exactly. So at repeated time scales. And there's an effort to include a wide cross-section of the population and ensure diversity and representativeness of the data. The objective is disease prevention, understanding health trends, and personalized medicine. Within the appropriate ethical and privacy concerns, so that we can have the management of the data in a way that complies with the wishes that the individuals sign for in terms of the usage, that the management of the data is properly handled, ensuring its quality and its accessibility for further research, and that the ongoing participant engagement is active so that it benefits the people who are supposed to represent those data sets rather than the big corporations. So examples of population-based biobanks, we have the UK Biobank, which contains 500,000 UK participants. We also have the All of Us program, which is in the US, aiming to gather data from 1 million or more people living in the US. And the Estonia Biobank, which apparently includes something like 10% of the whole population of the country. Disease-oriented biobanks, they collect individual samples from individuals diagnosed with specific diseases or conditions. And the purpose is to facilitate in-depth research into specific diseases so that we can uncover mechanisms, improve diagnostics, and develop targeted treatments. That means that we target collections, so patients affected by a particular disease within clinical settings, detailed clinical data, treatment responses, outcome alongside biological samples, and then usually there's close collaboration with hospitals, clinics, so that recruitment is ensured. Again, I'm going fast here because this is just to, I'm not going to examine on this. I'm just giving you a little bit of introduction, hopefully slightly less, how can I say, basic, perhaps, hopefully. And I'm going to go back. So we want to enable research, the study of biological and the genetic foundations. We want to facilitate, for instance, the effectiveness of various treatments, or we want to identify biomarkers that can predict disease presence, etc. What's a biomarker? Very good. Yeah, that's a very good definition. So challenges, recruitment. How do we make sure that the research design is actually inclusive? That's one of the biggest topics right now. How do we make sure that what we call here postcode lottery doesn't affect you? Depending on where you live, you're going to have different access. So how do we minimize that? How do we make sure that sensitive health information is robustly kept? How do we make sure that we have appropriate and balanced disease progression and treatment outcome data sets recovered? An example of disease-oriented biobanks is the Cancer Genome Atlas, which comprises over 30 types of human cancer, Alzheimer's disease neuroimaging initiative, which contains Alzheimer's disease data through integration of clinical imaging and biological markers. And then another example could be the Parkinson's progression markers initiatives. There are other types of biobanks, like virtual biobanks that do not store physical samples, but provide digital access to biobank inventories. So it's a repository, really. So that sharing of biological material and data information is maximized. Tissue banks, genetics and genomic biobanks. By the way, I come from a genomic background, so you're going to get a lot of that, I'm afraid. But hopefully it's just a useful use case that is applicable to other types of data, all right? As I said, I cannot conceive modern biomedical research without the use of biobanks. So if you're gonna do any type of research on biomedical area, you're gonna use biobanks one way or another. And they support personalized medicine. Now, what's different about what we're gonna be talking here is that most of the biobanking courses, stuff that you look at elsewhere is around the lab side of things. You know, the how you manage the samples, how you collect the samples, how you retrieve the samples, physical. But in our module, the emphasis is going to be on the data. So all of that, I'm not an expert. I'm going to mostly ignore. I'm going to assume that there are many different colleagues out there that, you know, will provide for me the lab support, because frankly, that's kind of cheap. And where the actual demand is, as I said earlier, is on the interpretation aspect of big data sets. So because biobanks have become so data rich, we have a new set of problems. You know, how do we access? How do we share the data? How do we facilitate collaborations? How do we protect those sensitive data from, I what do you call it, from uses that are not legitimate, basically. How do we automate and track the systems so that we have the maximum capacity for usage? So this is going to be extended, and as I said, there's going to be many challenges and opportunities that we're going to discuss throughout this course. I've gone quite quickly, and I guess I'm going to give you my next presentation, which is more research based, and that's going to give you a little bit of a flavor of what real research at hopefully the sort of an international level feels like. And then after I give you that presentation, we're going to go back there. I'm going to stop the recording and we're going to talk about research. So as I said earlier, the presentation was given at King's College London last February. I had like maybe 200 people attending. Now, this is my own research, okay? So it's going to be around the things that I'm doing, obviously. Now, I'm not going to assume that you know the different concepts, that you know everything that I'm going to say. So I'm going to stop quite a bit just to make sure that, you know, we all understand the terminology. Now, can someone define to me what a large language model is? Magda? No? Daniel, what's a large language model? All right, okay. We're getting now into the new field. It hasn't taken that long, has it? Okay. Anyone wants to give it a try as to what's a large language model? Have you heard of that before? Right. Yeah, are you being a bit shy? Are you being a bit shy? Okay. All right. So I do a lot of research on that. Yeah. No follow. No? Okay. No? Tell you want to give it a try, or shall I give it a try? I'd rather you give it first. What's a large language model? Okay. Tell me. I love when you really try things. Okay. Good try. Good try. You're getting hotter. A large language model is a statistical representation of billions of words that have been scraped from the internet, from the research literature, from Wikipedia, from Reddit, whereby is able to map the likelihood of how a particular word relates to another. And it's basically a way to encoding relationships between the words as observed in the body of human knowledge, whereby you end up having representations of weights that basically describe how words relate to each other. That wasn't the best explanation. I thought I was going to explain it better than this, but as you said, large language models underpin ChatGPT, for instance. So in a way, except that ChatGPT is a large multi-model model, because ChatGPT not only uses language, it also uses images, it uses sounds, it uses multimodal data sources, okay? But what I want you to take away from this is that it is large because we are talking about, in fact, trillions. ChatGPT, they haven't released how many trillions of words they use, but I know this in the order of trillions of words. So if you were to take all the body of knowledge of the internet, process it in terms of understanding through a model called transformer, which you may or may not have heard, and assign specific weights to how, in fact, it's not words, they call it tokens, because tokens is slightly different, but I'm going to say just words. Basically, a large language model has cracked the way we humans build sentences. I mean, you see it on ChatGPT, it's mathematically cracked the most likely relation that exists between words. And you can represent them in a way that semantically retains the understanding of the context. So the big difference between, let's say, translators that we used to use with Google, you know, they didn't work so well because they could only model the relationships between the previous word and the next word. And what large language models are able to do is that they can actually model the relationship that a particular word has with the general context of the prompt you give it. Hey, this is new. How do you feel? You're all now poker faced. Now, can someone explain to me what large language models is. Mhm. Mhm. Okay. So how about if we use the power of existing large language models in order to ascertain how well they can perform at representing the information that is provided by biobanks such as the UK Biobank. Do you think that that's okay? How do you feel about that? Sorry? How about if we use large language models to understand how much they're able to represent the information contained in the UK Biobank? To predict. That's what this talk is about. I could have also entitled it as experiences in leveraging opportunities and excitement that exist in mining biobanks with large language models. Does that make a better sense? Not so much? Am I scaring you? This is supposed to be scary. Do you know why? Otherwise, it wouldn't be research. If you don't feel out of your breath, you are not doing research. Can you write that down? Or shall I write that down? Yeah? Would you agree with that, Itir? You've done research, right? Well, you're about to get a PhD at Ankara University. At Ankara's university. Does it, do you feel uncertain when you do research? You don't know what you're gonna find, right? You know, you might be lucky, you might not be lucky, you know, you research, search, research, search, research. What is research? And what are we searching for? Answers. For what? For a question. And guess what? The most important thing, I believe, no, I know the most important thing for a scientist, and you are scientists now, is to know how to properly ask the right type of questions. Would you agree? It's about asking the right question. So wouldn't it be nice to use large language models like ChatGPT to see how good they are at understanding biological knowledge that is encoded in resources such as the UK Biobank. So that's what this talk, research talk, is about. Any questions? Am I pushing you too hard? No? Am I? So we have a mind here. And you have the machines, the drills, they're going this way, that way. Imagine there's a massive pot of gold at the bottom of it. But you need to drill. You need to find it. You don't know exactly where it is. Imagine that this is the UK Biobank. Yeah, this is the oil of the 21st century. Data. How is the UK Biobank currently mined? Well, you have this researcher from all over the world. They make an application. They have an interesting question. They want to research. They apply for access. They get granted the access, and then they start drilling this way. You have someone in Ciudad de Mexico, Mexico City, and they have this question. They're very interested, and they start drilling this way. You have someone in Cape Town that is interested and start drilling this way, and it's gonna try to find something. You have someone in Tashkent that is interested in that question and start drilling this way. I would say that that's problematic because there's no coordination. And wouldn't it be nice to use large language models to help me ask the right question so that I can begin to query the most likely type of data that will maximize benefit. So that's the research I'm trying to do here. You haven't seen this, but this is me, September 2024, in the Amazon jungle, Peruvian Amazon jungle, collecting genome samples. Okay, so one of my biggest research contributions has been the characterization of the specific features, genetic features of indigenous genomics population, indigenous populations in the Amazon. That, and I'm still working on that, but that'll be the topic of a different class, perhaps another time. Here, I'm just going to focus on really what I want you. I want you to become experts at the UK Biobank, and we're gonna look at from many different angles. I can assure you that if you can come out of this MSc degree saying, I've got experience using UK Biobank data, I've used it for this particular research project, I can tell you that as an employer, I would be very interested in those kind of skills, because the UK Biobank is well known everywhere, and it is basically the sort of standard in terms of biomedical research. It's kind of the benchmark, global benchmark. So hopefully that helps. But I'm just using now the UK Biobank as a tool for research. Okay. So large language models to elucidate biobank-derived the insights into health and disease. And what I'm trying to do is to make sure that large language models can help us optimize the way we mine data banks. Wouldn't that be nice? So what are large language models and what are they good for? So large language models are AI systems trained on massive text corpora. So, you know, the stuff I mentioned on the internet, to understand and generate human-like language. And they use something called transformer. The transformer paper was published in 2017. And its title is Attention is All You Need. It's one of the most cited papers ever. So if you want to know exactly where the AI revolution that we see today started, that would be my starting point. Attention is all you need. So you search in Google, attention is all you need. Space transformer, you find that paper. It's been cited I don't know how many times, but that is the underlying theory behind the AI revolution that we live today. Is that helpful? Do you feel you're learning something new now? Yeah? Are you getting now into the not so comfortable zone? We are gonna get out of your comfort zone. That's the only way we learn, isn't it? Well, this is what you paid for. So LLMs model the relationship between words in context, allowing them to produce coherent, contextually relevant responses. So that's not too dissimilar from what I said earlier, hopefully. Yeah. And LLMs have been used for genomic data analysis. So here you have something called the EpiBrain LM, which, by the way, can someone tell me what ChatGPT stands for? What does GPT stand for? You are using it every day and you haven't even thought what that could be. You need to change your way of thinking. You need to start questioning things. Yeah? Generative pre-trained transformer. So you are using transformers every day. You didn't know about it. You are using large language models every day and you didn't know about it. I want you to know about it from now on. Okay? Because that is what is underpinning the AI revolution. we are all living today. So large language models have been used for neuroimaging, encoding regulatory and epigenomic signals, aggregating information from many different genetic variants at once, improving the power to detect genotype-phenotype associations. Right. Another set of words I want you to know. What's a genotype? Okay. Temu. No, forgot. And do. What's his name? Tunde. What's a phenotype? I want you to know that. Alina, what's a phenotype? Hair color. Susceptibility to Alzheimer's disease. Allergies. Height. Weight. What's a genotype? Yeah. And genotype? So you've defined really well phenotype. The one that is not so well defined is genotype. What's genotype? The genotype, the way I like it to define it in broad terms, genotype is what you see at the level of the sequence of DNA. In other words, it's a collection of specific genetic variants associated to a particular feature, such as eye color, hair color, diabetes. Okay, so illnesses are phenotypes. Variation in genes are genotypes. Okay? So the LLMs have been used for that. LLMs have been used for stratification of patients. What is stratification of patients? Sir? Yeah, classifying patients according to a set of criteria as designing an algorithm that gives you clusters, for instance. Yeah. So you can stratify patients based on their genetic similarities. You can stratify patients based on 10,000 features from the UK Biobank each one has. They've also been used for modeling risk. Now, let's go back to basics. Biobanks. So they were initially designed as static repositories storing biological samples for research to further understand health and disease. Why we need biobanks? Because they allow sufficient data quantity in order to find statistically significant relationships. In order for us to understand biological processes, we need many samples. A lot of the time, the signals or the markers are so subtle and the changes are so subtle that small numbers don't do. Same as, you know, if you think about large language models, we've needed to have all of this of the internet for it to be able to model, have enough information to be able to model human language in a way that sounds natural. Here you have the UK Biobank summary of different types of data that it contains. It contains physical measures, lifestyle, cognition, biomarkers, imaging, online service, physical activity, genetics, and so on. Here is a global map of banks from around the world. As you can see, the representation is not very balanced, should we say? Alina, what's happened there? Daniel, what's happened there? What's happened there? Why is it in color? Well, there's a federation of biobanks, African biobanks, not necessarily contain much Nigerian information, but it does contain African information. I think it's a little bit misleading. And what's happened there, Nofo? Okay. Iti, what happened there? Don't know. And there? Oops. Nothing. And there? Okay. It's nice to see all these global representation that we have in this class, in this MSc. So the problem is really that we have scattered, biased, siloed, heterogeneous data types, format types, difficult to access, difficult to understand the structure. Even for the UK Biobank, data is messy. If you log into the UK Biobank, this is what it looks like. It's very difficult to really understand what the contents are. Even if you have access, it takes a long time. You have to understand the tool that is being used, in this case, DNA Nexus. And it's incredibly difficult, even for me as a bioinformatician, to get access and to really perform the analysis is very challenging, let alone for someone who's trying just to get into the. How about the UK Biobank? Is it representative of the UK population? So there are more women, older age groups, and fewer people from less socioeconomically deprived areas. Subjects are less obese, less likely to smoke, drink less alcohol, and have fewer self-reported health conditions than the average UK population. And the linked health records show fewer rates of all-cause mortality and total cancer incidence than the general population in the same age than the UK. In other words, the people who are participants, they tend to be educated, wealthier people, white people. Even though the UK is meant to be sort of diverse country, here you have the ethnic background of UK biobank participants. 400 of the 500,000 that they have, 472,000 describe themselves as British, 14,000 Irish, any other white background. I suppose I would myself be any other white background. White, Black, Caribbean, Asian or Asian British, Indian, Pakistani, Bangladeshi, Black or Black British, Caribbean, African, any other background, Chinese. So it looks something like that. Do you think that's problematic? Why is it problematic? Mm. Mm. Yeah. Yeah. I'm not saying it's not useful. It is useful, but is it representative? Mm. And to be honest, at the time when the UK Biobank started, the UK population, 95% was white British. So in fact, it's a healthier snapshot of what used to be the population 20 years ago. Now it's a lot more diverse. And to be honest, the UK Biobank, even though it's as skewed as it is, I think it has been a tremendous force for good. Because even though the representation is skewed, there's no country or researcher they're gonna deny access as long as they are legitimate to do their research. And I believe that access to that data to maximize research, as long as you provide some guarantees that that is going to be properly handled in order to protect the rights of the individuals to be anonymized. I think that's a tremendous process for inclusion. So I want to also say that even though that's the case, that the data is very skewed, you know, I'm not blaming them. All of these things, we're just now beginning to learn about these kind of things. I mean, it's just a snapshot of the UK population. I mean, so yeah, of course, most people are white British, because the majority of people are white British. But I think let's point out the where the gaps are so that we can address them. Because at the end of the day, you know, as a researcher, as a health equity advocate, which is what I base my whole research on. I've lost track of time. What's the time? We have half an hour more, right? So as a health equity advocate, I cannot conceive precision medicine or personalized medicine or the medicine of the future without having health equity at its core. Because the bias on the data sets does not only affect the underserved populations, it also affects the people who are being served, because a lot of what is captured as signal is actually noise as a consequence of not having the appropriate representation. And one of the big arguments I'm trying to do is that actually a lot of our knowledge is incorrect precisely because of that skewed representation that we have, because we are representing false positives that shouldn't be there as a consequence of the lack of diversity. So for white people and from as well as for non-white people, it's tremendously important to have the representation of the whole of the human race. Now, I've said the word race. That will be the last time I'm going to say the word race. I don't like, in fact, I don't like to say even ethnic, because they're all socially conditioned labels. I prefer to talk about ancestries, in the sense that genetic distance genetic differences between groups of populations, which are fluid, which are not really discretely separated. It's all a continuum. But those ancestry differences condition different populations' abilities to metabolize drugs, to respond to diseases, and to have successful treatments. So hopefully that's kind of clear. And I totally am against this classification, because it's purely based on socio-geographical rather than biological, socio-geographical and or historical contexts, which are inherently unequal, inherently unfair, I would say. It's getting interesting now, isn't it? You didn't expect that, did you? Well, you expect anything now from me, actually, at this point. So Can you see how it's all intertwined? You know, biology, politics, economics, history, and the data that we collect, analyze, reflects that. And unless we actively work on debunking entrenched historical biases, in the AI revolution, these biases, unless we actively try to work on them, they're going to be augmented, amplified, exacerbated. They're going to. You know why? Because there's a lot of bias there that we don't even know about. And my mission as a scientist is to make sure that no patient should have any different diagnostic or treatment, regardless, simply because of who they are and where they come from. That's my sworn mission for the rest of my career. You know what the sad truth is? That there's no money in that. That the current politics, the current world where we live in, the only thing that matters is money. And until and unless we have viable, profitable business cases where we can actually make money on making things more equal, this equity aspiration is just going to be a dream. That's my current thinking. I'm not sure if you agree with that, but you can challenge it. But I can tell you, we need to find a way to make money making health more equitable. Otherwise, the current differences are going to be exponentially augmented. You know why? Because the people who are making the decisions on how we classify those labels, on how we group people, have no idea about the context, the history, the culture of the people they are analyzing. It has never been so urgent than today that we are able to find ingenious ways to bridge these gaps. So I hope that you can help me throughout this year pulling towards that direction, because that's what my research, that's what I would like you to think about. And hopefully I'm just one person, but you know, if we're able to all pull in the same direction, we might make it 0.1% better. If we could make it 0.1% better, you know, it'll be worthwhile, because that will compound. And then the next year, it'll be 0.1 better of, you know, 100%, 0.1%. And it, you know, that's how it it gets better. So back to the UK Biobank. So my master student from last year, he counted how many different features the UK Biobank has. We counted 14,000. In other words, there are 14,000 different types of measurements in 500,000 people. You know what my definition of big data is? Big data is when the data you're handling cannot be put on an Excel spreadsheet. What do you think, Iti? Do you agree? Last time I tried, when you get onto 30,000 rows, what happens if we have like some tables I've been dealing with 128 million rows? Or like one of my experiments that I'm doing right now, where I'm analyzing 10 million publications. I'm afraid, ladies and gentlemen, you want to do anything exciting, you have to be in the order of millions. That's the age we are in now. Seven years ago, 20,000, yeah, around that. You want to have impact, you have to get into the millions now. You have to be a millionaire. So this is just a representation of the type of data that the UK Biobank has. This is the evolution of the questionnaire data that the UK Biobank has been collecting. Diet, cognitive function, occupational history, mental health, digestive health, chronic pain, food preferences, cognitive function, mental well-being, health and well-being, visualization and memory, repeat chronic pain. It's pretty heterogeneous, isn't it? I bet you any kind of disease that you would potentially be interested in is somehow, somewhere contained in the UK Biobank. And if not, it's probably not even worth it going into that. You know what the problem is? That, and I have that in a different presentation, which I'll give it to you if maybe not, I'll see, depending on how you behave. I will many of the diseases that affect what I call the global south are missing. Things like tuberculosis, malaria is almost absent. What other diseases? Some of the most deadly diseases that kill most people are not present in the UK Biobank or any of the biobanks that exist today. Simply because the vast majority of all the data that we have is led and contained by countries in the global north. And I would say that the UK Biobank and the UK in general, I think I did say this at the beginning of the last week, that what the UK has is that concentration of massive biobanks that nowhere else in the world is there. So I think that's a tremendous edge that we'll enjoy just by the sheer fact that we are here. I've been talking non-stop. You guys have any questions? How do you feel so far? Is this more of what you wanted? Okay. So if you actually go and look on the UK Biobank, it's incredibly complicated and massive. And access is costly, takes time. This idea of democratizing access to the data, very important. So I wanted to understand the effectiveness of large language models in retrieving and summarizing key biomedical insights from UK Biobank related literature. That's a very long-winded introduction for my research in order to get to the end. So that's exactly, that's the, remember we were talking about search, research. So what's the question? The question here I'm asking is, how effective are LLMs at retrieving and summarizing key biomedical insights from UK Biobank related literature? Shall I repeat? How effective are LLMs in retrieving and summarizing key biomedical insights from UK Biobank related literature? And then I have 3 objectives. So for your research project, this is the most important slide that you should relate to. Can you take a picture? We're going to spend a lot of effort and a lot of energy and a lot of time refining, iterating, defining, writing, rewriting, editing our aims and objectives. What do you think, Priya? Okay, sorry? Nervous? I don't bite. I am very, very easily contented with the very best. I didn't say that. I'm quoting Churchill. He used to say that. So I'm easily contented with the very best. I have a lot of patience, infinite patience for those who work. I don't care if you make mistakes, but if you don't work, I don't have a lot of patience. I'm not gonna give you favors. Yeah. And I'm not gonna, if you don't work, I'm not gonna give you a good reference letter. It's up to you. I'm not here to waste your time or mine. So I'm not trying to scare you, okay? I'm not gonna, I just want you to try hard, really. I just want you to be successful. And there are no shortcuts. So basically what I did was to compare ChatGPT, Claude, Gemini, Mistral, Llama, DeepSeek. Have you heard of those? Alina, can you explain to the people who have no head what are these? Mm-hmm. Yeah, yeah, yeah. Does that make sense? So the UK Biobank has a website called the UK Biobank Showcase. And the UK Biobank Showcase has, you can actually download. I think I did show you last time, didn't I? The showcase, where I showed you, you know, how many individuals, some kind of statistic. So you can actually download descriptions of the contents of the UK Biobank through the UK Biobank Showcase. It's not a warning, but a note. We're gonna be, so I'm going to expect you guys to do research on the UK Biobank Showcase, because this is one of the best ways to really understand the contents, the thinking, the design, and at the same time learning doing things based on that, because we'll be doing bioinformatics analysis using showcase data. In this particular case, table 19 contains a massive file that summarizes all of the papers so far that have used the UK Biobank. $9,000 in total. So I'm using the knowledge contained in the 9,000 papers as the benchmark to compare against what the Mistral, ChatGPT, Gemini know. Okay? And I'm gonna ask all of those different LLMs these 4 questions, right? So I'm going to, all of them, they're gonna ask them the same question, and I'm going to analyze what results they give me. So the first question is, what is the subject of the most commonly occurring keywords in the UK Biobank papers? Second question, what is the subject of the top most cited papers relating to the UK Biobank? Third question, what are the top 20 most prolific authors publishing on the UK Biobank? Fourth question, what are the top 10 leading institutions in terms of number of applications to a UK Biobank? So these questions have a double purpose. On the one hand, is to first understand how these LLMs work, but actually my hidden purpose is to really understand the contents of what the UK Biobank has, which nobody, as far as I know, has published on before. So people are using the UK Biobank without really understanding, what's the impact, how is that really contributing to knowledge? And that was the sort of research question that I wanted to address in this particular piece of work. So here you have a breakdown of the total number of publications per year. So I haven't counted 2025, but because obviously we haven't finished yet, but more than 2,000 papers are being published related to the UK Biobank, which is way above any other type of research from any other database from around the world. So this is new. This is actually state-of-the-art research. It's not even being published. Well, I mean, it's in the process of being published as we speak. But this is bleeding cutting-edge research that on your first lecture you're being exposed to. Yeah? Hopefully you're getting value for your time and money. You can see here the subject of the most commonly occurring keywords in the UK Biobank. Humans, female, male, middle-aged, United Kingdom age, risk factors. Now you are getting down into the table and it's getting slightly more interesting now. Genome-wide association study. Polymorphism, single nucleotide. Mendelian randomization analysis, phenotype. What's a genome-wide association study? Genome-wide association studies is the workhorse genetic model for complex diseases. So most diseases that you know of, Alzheimer's, diabetes, cardiovascular disease, cholesterol, they all involved many genes. Genome-wide association studies capture SNPs, so genetic variations from across the whole length of the genome that are associated to higher risk of that particular disease or phenotype. Yeah, in this context, phenotype and disease is the same thing. So genome-wide association studies provide a collection of markers that have been associated to the presence of that disease. So if you have that marker, then you are more likely to develop that disease. And as I say, it's the workhorse tool for associating genetic risk for specific diseases, most of which are polygenic, involve many genes. Any questions? I want you all to understand this concept because it's going to be important. Any questions? What's a genome-wide association study? It's a collection of genetic markers who have been associated to a specific condition. And actually, I got it wrong. So this is the output of genome-wide association studies is that. But the but genome-wide association studies is actually is really the statistical methodology behind the production of markers from many different genes associated to a specific condition. Okay, so it's really a statistical model. All right. So from the top 20 keywords, apart from the trivial ones at the top, things are starting to emerge. Big number associated to genetics. So what I've highlighted there, they're all terms associated to genetics. And then you see, actually, even genetic predisposition to disease. I don't know why I have not highlighted that one. Prospective studies, cohort studies. So these are keywords. These are the most frequent keywords in the body of knowledge that has been output from this massive body of biomedical research. I would say there's a very strong bias towards genetic related stuff. And a little bit on, you can see cardiovascular diseases, diabetes mellitus, and brain, which I would contend are mostly highly industrialized types of conditions we would be interested in, which is where the money is. If we look at the most cited papers where they have been published, from the top 10, 50% have genetics or genomics in their journal title. If we look at the people who have published the most, we have George Davis Smith at the top, who is a professor at the University of Bristol. And guess what? He's the inventor of something called Mendelian randomization, which is actually one of the keywords somewhere here. So one of the methods for analyzing genomes, he developed it, Mendelian randomization, and so the guy appears at the top. What are the institutions that are publishing the most, that are accessing the most the UK Biobank? I'm singling out King's College London because, as I said, I gave this King's College London. But you can see the institutions that have requested the greatest number of access applications. Interesting, isn't it? Only in the top 10, the one at the bottom, which is Korean. The rest UK based. How are we doing for time? I think we're gonna leave it here. Maybe we can continue tomorrow. I'm going to stop now the recording.